Hey everybody, and welcome back to another simple science video. My name is Matangi, and today we're going to be starting the new A-level biology revision series, and this is just part 1, and we're going to be approaching the most basic topic in biology, which is microscopy. So let's continue. So just some basic knowledge, which shouldn't be really any surprise, but just a reminder, I guess. So microscopy is the study of cells, right? So first, let's see. Atoms and molecules make up cells. Cells, in turn, are known as the basic units which make up living organisms, such as humans, plants, fungi, viruses, etc., etc. Now, it's no surprise that most cells and their structures can only be seen using a microscope because they are teeny tiny. They're in nanometers and micrometers, and our eyes simply cannot cope with that. And just another tip, a small tip for people who are doing AS biology and in particular have practicals, definitely make sure you get your hands on a light microscope for practicals because it is super useful. You're definitely gonna need to use a microscope sometime soon, so just pop into your lab and try to get your hands on one. So let's carry on with the next slide, which is about the first kind, which is about light microscopes. So the first kind of microscope which we'll be looking at today is the light microscope, which, no surprise, uses light rays. And if you've seen the word photomicrograph, it simply means an image which is produced by one of these guys. Now, you've probably seen this in your school lab and you should be familiar with it. I don't recommend remembering all of these labels, maybe just the main ones, such as head, eyepiece, definitely stage, base, coarse focus, objective frames or objective lens. You will be told which lens to use in the exam, so I wouldn't worry about that. If you can see, I don't think you can, but there is a glass slide here, which you place the specimen on top there, and then you usually stain it. Back in the day, I used iodine. I don't know what people use nowadays. And then you have a cover slip to keep everything in place. And this is the illumination intensity knob, or in other words, the thing which basically controls the brightness of the light which is emitted from here. This is just a simplified drawing of how a light microscope works, and I'm just going to briefly explain to you, which again you shouldn't need to know in detail, but you should know how it works generally. So basically the light source from here emits light, which is this, which travels through this thing called the condenser lens, and it hits the specimen. Now this condenser lens focuses it into a narrow beam so that it reaches the specimen, not all scattered. And then after it reaches the specimen, it travels through the specimen, and the objective lens is your main friend here. This is the guy that does the magnifying, the main part of the magnifying. And then it travels through, it hits the projector lens, which also magnifies, but not as much. It just mainly makes it clearer, I guess, but you shouldn't really need to worry about that. And then it travels through and it hits the eyepiece, which again makes the image clearer. And it may be a magnification depending on what you have in here, but eventually the image is projected onto whatever receptor here, which could be the retina of your eye, or it could be a camera or a computer or anything. So it basically depends on the light which travels through the specimen, which is then magnified. The image is then magnified, the light rays, and it travels through and it should be received on whatever's on the other end. Alright guys, and now we're going to move on to the second type of microscope, which is known as the electron microscope. And I wonder if you could guess, it uses electrons. So instead of using light, it uses electrons. And an electron micrograph, again, is just an image which is produced by an electron microscope. Now this isn't actually an electron microscope, it's just a simplified diagram for me to try to explain roughly how it works. Now basically, it uses beams of electrons, and what that means is there is an electron gun and an anode. So when a metal is heated up, for example, it releases electrons. Now these free electrons, they form a beam, all right? So the electron gun and the anode, they form an electron beam, which then enters the cylindrical looking thing, which is a vacuum, by the way. Keep in mind this is a vacuum, and I will explain why in just a bit. So as the beam is traveling through, it first hits something called the condenser electromagnetic lens, which basically is like a guide, and it just simply directs the electron beam onto the specimen, right? The specimen is over here laying on a grid. So the specimen is placed on a grid, and then there's something called the objective lens, which does the main work. It's what magnifies the image. So that's your main guy in the electron microscope, right? And then as the beam travels through or travels past the specimen, it then reaches this thing, which is called the projector electromagnetic lens, which basically focuses, it's your focuser, right? Like the fine tune on the light microscope we saw, this guy is what focuses the image onto the screen. Now the screen at the bottom over here, it's a fluorescent screen. And the electrons, when they travel through, when an electron hits the fluorescent screen, it has chemical reaction, and the fluorescent screen in that area which has made contact turns white. So the parts which the electron doesn't hit stays black, and the parts which the electrons do hit stays white. So it kind of produces like a reverse X-ray, I guess. Not a reverse, just something like an X-ray with a black and white image. And again, whatever device is attached on the end, so like computer, records the image, right? So it records the black and white image, and then usually stains and like color graphics are added to clarify the image. All right, now let's get to why I mentioned that it needed to be in a vacuum, and now let's get on to the next slide in which I explain why. So first off, the specimen has to be thin, and okay, wait. Let's do it this way. It has to be in a vacuum. Why does it have to be in a vacuum? Because as I said, we're working with electron beams, right? So when an electron beam is traveling, if there are a lot of air particles, it's going to collide and it's going to scatter, and you're never going to get a clear image or a sharp image. So it defeats the whole purpose. So it definitely has to be in a vacuum. And for that reason, the specimen also has to be dead, because the water boils at room temperature. So therefore, if you're in a vacuum, the specimen's dehydrated and it has to be dead. So these three main things is what you need to remember for an electron microscope. I guess these are the disadvantages over a light microscope. And next, we're going to be looking at the two types of electron microscopes. Just like there are two microscopes, the electron has sub-microscopes. I guess you could divide it into. So let's see what those are. And the first type is called the transmission electron microscope, and this is the original type of microscope. So basically, the transmission electron microscope is the microscope which allows electrons to pass through. So you could see inside the cells. And the scanning electron microscopes is the ones which basically collects the reflected and rebounded electrons off of the specimen. So basically for this one, the scanning electron microscopes, you have a 3D structure of the outside surface of a specimen. Whereas transmission electron microscopes, you see inside the specimen. And for this one, the specimen has to be thin. And for scanning electron microscopes, it's better. It has an advantage over TEM because you get to see greater depths and you get to see the patches on the surface. I guess you get to see the structure of the surface, and it produces a 3D image. However, the disadvantage of a scanning electron microscope is that its resolution, which we will get to in the next couple of slides, is not as good as the transmission electron microscope. So just keep in mind that the transmission electron microscope is the one that transmits. So I usually like to think it as TEM is the one that's T for through, and scanning electron microscopes is just the other one, which collects the reflected beams of electrons. So it produces a 3D image. And guys, just another note, the amount of resolution which you can get with an electron microscope is much, much higher than what you can get with a light microscope. So that's an advantage of an electron microscope, and that's the main reason why electron microscopes are used, is because of their resolution capacity. And another thing is that you don't need to keep constantly focusing it like a light microscope. It would all focus at once. The whole entire piece of the specimen would focus at once, whereas the light microscope, you would have to keep tuning it up and down, right? So just keep those advantages of the electron microscope in mind, and keep these two in your mind as well. And with that, guys, we're going to be moving on to something called magnification and resolution, which is very important. But before that, we're going to have to cover this thing called units, which I personally despise. In the beginning, I found it actually kind of difficult, but in the end, you just have to, I feel like, memorize these 4 bullet points, this chunk of information, and you should be good. And these are the main units which you will be working in. Millimeters, micrometers, nanometers. Now, 1 millimeter is 10 to the minus 3 meter. 1 micrometer is 10 to the minus 6th of a meter, so a millionth of a meter. 1 nanometer is 10 to the minus 9 of a meter, so I guess 1 billionth of a meter. And to convert from millimeters to micrometers, so this guy, you need to times by 1,000. So to convert from millimeters, you times by 1,000 to get micrometers. And just really, I strongly recommend you remember this, because when you work in magnification, you want everything to be in the same units, whether it's millimeters or micrometers. Now, keep in mind to do the reverse. So to get from micrometers to millimeters, we are going to have to divide by 1,000. So all in all, before we hit the next slide, just take a minute, pause, and make sure you remember this very clearly, because these are the main units in biology for magnification. And now we can officially move on to magnification. Now, what is magnification? For all you biologists, definitely just remember this definition. It could get you marks. Magnification is the size of an image divided by the size of an actual object. So basically, in simple terms or simple language, it's basically the zoom. How zoomed is the object? So that's simple language. Don't write that in your exam. You have to write this line over here. The size of an image divided by the size of an actual object. Or in other words, if that doesn't make sense, the number of times larger an image is than the real size of an object. So just take a moment, just take this definition in, and I'll explain to you better with a formula, because words are more confusing. So now let's take a look at the formula, which I feel is a lot easier to explain. This is basically the formula triangle, which I recommend remembering. How I remembered is MIA, so the name M-I-A, MIA. M stands for magnification. I stands for image size, so the size of the photograph or the electron micrograph. And A is the actual size. And since we're dealing with cells, always remember that your actual size should be small. So if you get something like, I don't know, 1,000 millimeters for a cell, you've definitely gone wrong somewhere. And actual size, usually when they give it to you on a piece of paper, it'll be in millimeters. You'll usually measure it in millimeters. And I think that it's better if I carry on with the example. So let's check that out. So, measuring cells. First off, I cannot stress enough, you have to remember to convert to the same units. It makes life so much easier. Now, in the first case scenario, you are either told 2 of the 3 things. So for example, if you are told that the image is magnified by 100 times, and the image is 50 millimeters, right? So the image is 50 millimeters. So you have M, you have I, and you do simple basic maths to get A, which is the actual size. And the actual size is 0.5 millimeters, which makes sense because it's a lot smaller than the image, because the magnification is 100 times larger. If you're getting a number in the same unit which is larger than your image size, but it says it's magnified, you know you have gone wrong somewhere, so double check your answer. All right? So we're going to move on. And also, before we move on, you can be given any 2. So in this case, you were given these 2. Next time, you could be given the actual size of an object and the image, or the actual size of an object and the magnification, and you're asked to find out the image. So it's just simple mathematics, and you just cover up the triangle and rearrange a simple equation to get whatever you need. All right? So now we're going to check out the second scenario. So in the second scenario, which I find is particularly, I guess, harder to grasp, but it's still simple, don't worry. They give you scale bars. Now, if you've never seen a scale bar, it's pretty much a line which is scaled up, and they give you how much that line actually stands for, right? So here's how you approach a scale bar situation. You take your ruler. First thing you do, if you see a scale bar situation, you take out your ruler and you measure in millimeters. Now, in this case, we're going to pretend this thing is 3.6 centimeters long, so it's 36 millimeters, right? So in this case, 36 millimeters represents 6 micrometers. And the next thing you do, you convert millimeters to micrometers because the unit they have given you is in micrometers, so why not make it simple, right? So 36 into 1,000. If you don't remember on the previous slide, how do we convert from millimeters to micrometers? You times it by 1,000. So you get 36,000. And then to find out the magnification, you simply do 36,000 micrometers over 6 micrometers, and you get the magnification of 6,000. Now, why is it 36 over 6? It's the image over the actual, right? So the actual, just remember, the actual is the number which they give you, and the image is the number which you measured and converted, right? Now, this thing, or you could also do it this way, which is length of the scale bar divided by length that the scale bar represents, right? So the length of the scale bar here is 36 millimeters, aka 36,000 micrometers over the length that the scale bar represents, which is 6 micrometers. So you do the math and you should still get into 6,000 magnification. And if you get a fraction of a magnification, I think that you have gone wrong somewhere, because usually magnification is to make images bigger, not smaller, so you might want to recheck your math. But just basically remember this green part, or just remember that whatever number you measure, take it over the number which they give you. So that's how you approach a scale bar situation. And now another situation, excuse me. The next situation we have, guys, is using an eyepiece graticule and a stage micrometer. Now, an eyepiece graticule is basically a bar, which you will see on the next slide. Actually, you know what? I'll show you right now. Here we go. This diagram shows a stage micrometer with divisions 0.1 millimeters apart viewed through an eyepiece containing a graticule. So we're looking, this is the microscope, and we're looking through. And here is the stage micrometer. Now, the stage micrometer is basically what you use to calibrate these numbers, because these numbers don't have units. So you usually have a cell or something here. You place it here. So for example, my cell width goes from 10 to 50, right? So my units, my arbitrary units are 40. And 10 and 50, so 40 arbitrary units line up with one division on the stage micrometer. And one division on the stage micrometer is equal to 0.1 millimeters apart. Let's go back to the previous slide. Oh, my bad. Oh no. The divisions on a stage micrometer are usually 0.1 millimeters or 0.01 millimeters. So it's just a simple case of lining it up. So you know that the space in between here is 0.1 millimeter, and you know that, these units, they have no numbers. So for example, if your image goes from 10 to 50, you know that that's 0.1 millimeters. So it's just a case of simple ratio. And now, guys, earlier I mentioned something about resolution, right? So let's take a look at what resolution is. Now, resolution, to put it simply, is the ability to distinguish between two separate points. Now, magnification is unlimited. You can magnify to an image as much as you want, but it becomes pointless when you can't distinguish between 2 points, and the 2 points just appear as one. If 2 points cannot be resolved, they will be seen as 1 point. So no matter how much you magnify it, you'll just see 1 point. You cannot see further details. So the whole point of resolution is for detail. So the greater the resolution, the greater the detail. And which is why I told you earlier, electron microscopes, the main advantage they have over light microscopes is the detail. And let's see how that works in the next slide. This is an electromagnetic spectrum. For those of you who do physics, this should be familiar with you. And we are looking at the visible light region because we're talking about light microscopes at the moment. You can see that the wavelength of light goes from visible light goes from 400 nanometers to around 700 nanometers, right? That is the wavelength of light, of visible light, which the light microscope uses. So, visible light, it goes from 400 nanometers, as we saw, to 700 nanometers. Now, if, for example, you have a mitochondrion which has a diameter of 1,000 nanometers, which, for example, this is the mitochondrion. This has a diameter of 1,000, let's just pretend that's a circle, okay? It has a diameter of 1,000 nanometers, right? Which means it can interfere with the wavelength because this number is bigger than these two numbers. It's bigger than 700, so therefore, you see the wavelength, it interferes because it's too big. So therefore, you can see it and you can distinguish between mitochondrions because they're big and fat, and it doesn't fit in between these gaps. Whereas, for example, ribosomes, which have a smaller diameter, which, for example, let's say 25 nanometers, it cannot interfere with the wavelength because it just fits in the gap, right? So basically, it squeezes in the gap, so it's undetected. So therefore, it cannot be seen. And let me tell you about the rules. The general rule is that the limit of resolution is 1/2 of the wavelength of the radiation used to view the specimen. So what that means is that microscopes using visible light, so for example, the lowest wavelength of visible light, as we saw earlier, is 400 nanometers. So for light microscopes, divide that by half. 1/2 of the wavelengths, which is 200. So therefore, the maximum resolution for a light microscope is 200 nanometers. But however, the electron microscope, the wavelength is so much smaller. Therefore, you can see in so much more detail. Oh, another thing about light microscopes, transparent objects, they need to be stained. If not, the light will just travel through and you cannot see anything. So do keep that in mind as well. And now we move on. Electron microscopes, as I said earlier, use free electron beams, which behave like electromagnetic radiation, right? Now, the electromagnetic radiation of the electron beam, let's just say it pretends like a wave, right? Like the waves we saw earlier in the electromagnetic spectrum. However, the electron beam, compared to light, has a much shorter wavelength and therefore a lot more energy. So therefore, if it has a much shorter wavelength, if we go back, just imagine the gaps between these lines would be so much smaller. Therefore, it could detect something like the ribosome with 25 nanometers, right? Whereas if it's light, you cannot detect it. But if it was an electron, the gaps between these lines would be so much smaller. Therefore, you could definitely detect the ribosomes. So I'm sorry if I keep going back and forth and it makes you dizzy. And so what you need to remember here is that electron microscopes are suitable. Electron beams, in particular, are suitable because they have a short wavelength and because they're negatively charged. And why is being negatively charged, why does it make it suitable? It makes it suitable because you can focus the electron beams easily using the electromagnets, and therefore you can alter the path easily, right? So another interesting fact, the electron microscope has a resolution up to 400 times better than a light microscope, and the resolution can go up to 0.5 nanometers. So that's the end of the first chapter, guys. I tried my best. And thank you for watching, and make sure you check out the other videos of Simple Science. Thank you so much, guys. Bye. Hello everybody, and welcome back to another Simple Science video. Today we're going to be carrying on the A-Level Biology Revision series, and this is part 2, and today's topic is on cell structure. So let's get started. The first thing which we need to do is to distinguish the types of cells. Now, it can be categorized into two main groups. The first group is prokaryotic, which means no nucleus, which means the DNA is lying free in the cytoplasm. There's no nucleus structure. And the second group is eukaryotic, which means it contains a nucleus. Now, a common example that you know of a prokaryotic cell would be bacteria. And eukaryotic cells, common examples you would know is fungi, animals, so you and me, plants. Now, protista and protists are the same thing. It's just singular and plural nouns. What I mean by unicellular, so you and me were made up of a lot of eukaryotic cells, whereas protista or protists organisms are only made up of one eukaryotic cell. Another thing to keep in mind is prokaryotic cells are much smaller than eukaryotic cells. And the main feature for them is that their DNA lies free in the cytoplasm. Now, before we move on to see what features distinguish between these two, let's go and check out what features a prokaryotic cell will have always and sometimes doesn't have. Alright, so now let's look at the prokaryote cell features. Now, for your exam, I would just recommend remembering this column. Now, if you see that the number one hint that it's a prokaryotic cell is that if the cell wall is made up of murin, which is a peptidoglycan, which is a compound. Now, if it has these features, so cell wall, cell surface membrane, cytoplasm, circular DNA, and ribosomes, then it could possibly be a prokaryotic cell. So just keep that in mind. Now, additional features are just flagellum and infloating. This CSM is just cell surface membrane. I just abbreviated it. A capsule for protection, plasmid, and pili for attachment. Now, that's not here on this diagram, pili, but it's just when bacteria, they want to attach to another cell, then they just have extensions. So that's what pili is. So for your exam, just remember this table, but more specifically, remember these points. Now let's just quickly check out viruses. Now, viruses aren't really considered living things, therefore it wasn't in the cell chart because they do not do the 7 things. So Mrs. Nerg, they don't carry that out. Now, these are some important notes on viruses. First thing is they do not have a cell structure and they're not considered living, as I said, because they do not carry out the 7 things, the 7 living Mrs. Nerg concept. And more importantly, they do not have a partially permeable membrane. Now, here is just a simple diagram of one of the most simplest viruses. Viruses, they work by taking in a host cell and then taking over their ribosomes to create more of themselves. Therefore, they can't really replicate unless they're inside a host cell, which is one of the reasons why they're not considered living. So, yeah, just a very simple structure of a simple virus. Alright, now let's move on to the main topic, which is animal cell structure. Now, this is just a little content or background information. We have to use electron microscopes, which I covered in the previous video. So if you were unsure about electron microscopes, please do go check that out. And this is just a animal cell structure for reference, which I will be referring to throughout the next part of the video. Now, division labor, it means that each organelle has a specific job. Now, what is an organelle? Just like how we have organs in our body, like the heart, the lungs, the liver, etc., a cell has its own organs, which are known as organelles. Now, let's take a look at the largest organelle in the animal cell structure, which is the nucleus. Now, if you look at this diagram here, you can see that this is the nucleus and it is made up of certain things. So, nucleus is the headquarters of the cell, and I'll get to why that is in just a minute. First of all, the nucleolus, it's the darkest, densest spot that you see on the diagram. Now, why is it the darkest, densest spot? It's because it contains chromatin. Now, chromatin is simply loosely coiled chromosomes. So just keep in mind that the nucleolus is the darkest spot within the nucleus itself. And then we have the nuclear membrane. So the nuclear membrane is what surrounds the nucleus. as you can see, a nuclear envelope, also known as nuclear envelope. It is a double membrane, and just like cell surface membrane, it controls what goes in and out of the nucleus. Now, if you can see the little gaps here and there, it's basically nuclear pores, as it says here, and what that is just gaps for substances, big substances, which cannot pass through the membrane to go in and out. And specifically what substances, and here's a detailed, a more 3D diagram just to help you visualize of the nucleus. Now, you may ask, what substances go in and out of the nucleus? Well, the nucleus makes its own ribosomes and makes its own mRNA. And these ribosomes and mRNA are passed out into the cell. And why are they needed? They're needed for things such as protein synthesis, to help make nucleotides, to help make ATP, to help make hormones, and a whole bunch of things. And as I mentioned here, the nucleus controls the activities of the cell. So they're the headquarters, they're the boss. Now why? That is because the nucleus controls, the DNA inside the nucleus. Not all DNA is useful. Only some DNA is useful, and that useful part is called the genes. Now, the genes is what is expressed by the ribosomes and the mRNA, which is coded within the nucleus. And because it controls the things which control the proteins and things like that, it basically controls the activities of the cell and the inheritance of the cell. So the nucleus is the boss of the cell, and it's actually a 3D structure made up of these three things. Now let's move on to the second organelle, which is the endoplasmic reticulum, also known for short as ER. Now, what is the endoplasmic reticulum? So on the diagram here, I've made it bigger, is these foldy parts, as you can see. Where is it? Yes, rough endoplasmic reticulum, and then there's the smooth endoplasmic reticulum, which is basically, the endoplasmic reticulum is continuous with the outer membrane of the nucleus. So as I mentioned before, the nucleus membrane or the nucleus envelope is a double membrane, and the outer membrane is just continuous. So it's just folds and continuality of the outer membrane of the nuclear envelope. Now, it can be split up into two, and it has the rough endoplasmic reticulum, which I'll get to why it's rough in a minute. But basically the endoplasmic reticulum, if you want to visualize it, it's just an extensive network of sacs or just membranes and folds, and whatever reactions or whatever things that happen here can be separated from what happens in the rest of the cytoplasm within the rest of a cell. And basically, these sacs are connected to form an extensive network, and it's just like a parking lot with a ramp. So it goes zigzag, zigzag. As you can see here, zigzag, zigzag. That's what it is. Now, why is a certain section is called the rough ER? And it's rough because it contains these things called ribosomes. And we'll get to what that is in a minute, but just keep that in mind. And this is the main site for protein synthesis within the cell. Okay? And then the second part is the smooth ER, which, why is it called smooth? It's because it doesn't have these things called ribosomes, so therefore it's a more smoother and more flatter surface. And the function of the smooth ER is the synthesis of steroids and breakdown of substances such as toxins. And cisternae are simply folds of the membrane. So it forms, when I say form cisternae, it means when it folds, it's just the folding of the membrane, to be honest. All right, now mentioning ribosomes, let's move on to the next slide, which talks about ribosomes. As soon as you see ribosomes, you should link it to the site of protein synthesis. The function of ribosomes is the site of protein synthesis. If you want the mark, just write this down. Now, ribosomes are teeny tiny structures. They're only 25 nanometers, and they're made up of RNA and a protein. And you can see here on the diagram, ribosomes are these little black dots. So they're very, very tiny. And they come in two forms. They come in 80S ribosome and 70S. And when I say forms, I mean size. You can ignore these units on top. Now, these 80S ribosomes, which are the bigger ones, are found in the RER, so rough endoplasmic reticulum, and are just found free about hanging in the cytoplasm. And the 70S ribosomes are found in the mitochondria and also chloroplasts in plant cells. And I'll get to why this makes a difference in a minute and what theory it suggests actually. And the proteins which are made in the rough endoplasmic reticulum, they're first made in the RER, and as they move through to the SER, they are then modified. So it's basically like a process. First RER, the proteins are made, and then the SER modifies their proteins on their way throughout the sac. Alright, now let's check out the next organelle, which is, it has a lot of names, but I like to call it Golgi body because it's really simple, but it can be also known as Golgi apparatus, or it can be known as the Golgi complex. And basically what it is, it's over here. And basically what it is, it's just a stack of flattened sacs. Okay, and there can be more than one in an animal cell, but generally I think you'll just come across one during your A level, and we're going to keep it simple for our sake. And it's constantly being formed at one end of the ER, and it's broken down again on the other to form Golgi vesicles. So basically its process is it collects, processes, and sorts molecules ready for transportation. And an example would be, as I've written here, addition of sugar to proteins. So for example, to form a glycoprotein, or it can be the removal of amino acids to form a functional protein. Or for example, in plants, it can be used, the enzymes convert the sugars to a cell wall, so a functional, a structural component, which then will be used to build the cell wall of the plant. So let's go ahead and take an actual look at the process, which has 4 steps. Well, it's made here, written down here in 4 steps. So basically, in the first step, the proteins are made by the ribosomes in the rough endoplasmic reticulum and then are pinched off into vesicles. So it's made in the rough endoplasmic reticulum and then it's made into vesicles. So here it is, a transport vesicle. All right. And then the second step, the vesicles containing the proteins fuse into the Golgi body where the proteins are modified. So the purpose of the Golgi body happens. And then the third step, the vesicles containing the modified proteins break away from the Golgi body, so here. And then, so it's also known as the secretory vesicle. And then the fourth step is simply exocytosis. So the vesicle fuses with the cell surface membrane, releasing the proteins to the outside cell. So it's just a simple 4-step process, and I recommend you remembering this. Next, let's check out lysosomes, which are basically spherical sacs with no internal structure, and they contain hydrolytic or digestive enzymes which are responsible for the breakdown of old and unwanted organelles. And why is it contained in these sacs? It's because you don't want the entire cell to be digested, so it's contained in these protective sacs, and they're only used where they need to be. By the way, this is an animal cell. I just wanted an enlarged or zoomed version of a lysosome because over here you can see the lysosome is only a circle. And it's a single membrane, and it's around 0.1 to 0.5 micrometers. An example of where these will be used is in mammary glands. After lactation, they digest away the lactating cells, or lactating the organelles which are responsible for lactation, or another example would be, a more common example would be in white blood cells, where these lysosomes are used to digest the bacteria. And now let's move on to the next organelle, which is known as the mitochondrion. Now, as soon as you see mitochondrion, you should realize that this is the powerhouse of the cell. This is where all the energy is made and is released, and it's what keeps your body going. Now here in the diagram, you can see that the mitochondrion is this specific structure, and the function of the mitochondrion, so if you want the mark, is responsible for aerobic respiration. It provides energy for the cell. Energy is released from energy-rich molecules through a series of steps. Now, these series of steps you will learn, for example, glycolysis, etc., in A2, and you don't need to know that, but you need to know that it is a series of steps. The secondary function is synthesis of lipids, but this is the main function. Now, a rough estimate as to the size would be around 1 micrometer in diameter, and muscle cells, so cells which make up your muscle tissue, will have more of these mitochondria because they require more energy. Now let's take a look at the mitochondrion structure because it's very well suited for its purpose. Now, just like the nucleus, the mitochondrion is a double membrane organelle. So first let's look at the inner membrane. The inner membrane is more selective, okay? So it controls more strictly what enters and leaves the mitochondrion. The inner membrane makes these folds, and these folds are labeled as here as cristae. And now you may ask, why are there these folds? Well, it's pretty simple really, and I think it's a year 7 thing or something like that. It increases surface area, and therefore it makes the reactions which happens, more reactions to happen, therefore it's more efficient. So whenever you say like, why is there a cristae, you just say it's because it increases surface area, therefore it increases efficiency because it allows more reactions to take place. Okay, and then there is the outer membrane, which is more permeable. However, it does not allow H2O soluble molecules to go in. Therefore, it has these things called transport membrane proteins. The specific ones are called aquaporins, and it forms aqueous channels to allow the H2O soluble molecules to pass through into the intermembrane space. Now, I keep mentioning intermembrane space. Well, maybe not keep mentioning, but the intermembrane space is the space between the inner and the outer membrane, and it is a very important space which you will need to know in A2, but you still need to know this for AS, so you just need to know that that's the space between the inner and the outer membrane. Now, cristae, as I mentioned, are these finger-like projections, and then there's the matrix, which is the interior solution. Now, there are the other things called granules, ribosomes, and ATP synthase. Now, this thing is an enzyme which is needed for the respiration reaction. So, as I said, it's very well suited, so therefore it contains these enzymes within its matrix. And then it has its own DNA, and it has its own ribosomes, and it proposes a new theory, which we will take a look at in just a second. Now, just before we move on to the theory, just a small piece of information and I guess a summary. Mitochondria produces ATP. ATP stands for adenosine triphosphate and is importantly known as a universal energy carrier. And as I mentioned earlier, the folding of the cristae increases surface area and therefore increases efficiency. And here is just a diagram, I guess, of a summary of the respiration reaction, so the series of steps, but you don't need to worry about that at this level. So let's go ahead and check out the theory. Now, I keep mentioning this theory, so let's actually take a look at what this theory is. This is known as the endo-symbiont theory. Now, endo meaning inside, and symbiont actually has a long definition. It is an organism which lives inside another organism for mutual benefits. Now, in the 1960s, the scientists actually discovered that the mitochondrion was not an organelle. It was actually an independent bacteria. Now, why do they believe this? It's because it has these things such as DNA, ribosomes, and granules, and these things were used to sustain these organelles. I mean, these tiny things, I guess, are mini organelles for this mitochondrion, is what helped it sustain independently before it was adopted and integrated into our human cells. Now, the ribosome is smaller than our ribosome. So the ribosome size inside the mitochondrion is 70S, whereas the ribosomes which are lying free in the animal cell and in the RER are size 80S. So this is what led scientists to believe that the mitochondria were actually independent before they were colonized by us. Now let's move on to cell surface membrane. Okay, so the cell surface membrane, let's see if I can find it here. Okay, the cell surface membrane, okay, here it is. It's called the plasma membrane, but it's also known as the cell surface membrane. It's pretty much the bodyguard in an animal cell, so it controls what's let in and out by these things called protein carriers or transport carriers. So it's made up of this thing called a lipid bilayer, which you will learn again in A2. I keep saying that, but it's true. You'll learn about these things in A2 in more detail. And the main function is it controls substance exchange. It's around 7 nanometers, so it's super thin. And they describe it as a trilemma because you can see it in a diagram as two dark stained lines. So if you have a cell diagram, which you will come across, if you see the two dark stained lines, that's the cell surface membrane, which obviously outlines the cell. Don't just do any random line. And it's partially permeable. The next thing which we have is the microvilli. Now the microvilli are just these finger-like extensions. I guess if you want to describe it more specifically, it's just the extension of the cell surface membrane, and it has the same function as the cristae as I mentioned earlier. It increases surface area for reactions and therefore it increases efficiency. And we have microvilli up next, which are these fuzzy little things that you see here. And the microvilli's function is the same as the cristae's function, I guess. It increases surface area for reactions, and they're known as finger-like projections. So they increase surface area, therefore increase efficiency. So for example, if this cell were to line your gut, it would have more microvilli so that more food can be absorbed. Alright, so again, it's a very simple concept. We're almost done, guys, I promise. Now what we have is something called, oh, where'd that go? Here we go. Something called microtubules, and you can't see that on this diagram mainly because it's very small, but microtubules make up centrioles, which I'll cover on the next slide. So what is a microtubule? Microtubule is long, rigid, hollow tubes located in the cytoplasm, and they're very small. They're only about 25 nanometers in diameter, and what they're made up of is actin and intermediate filaments. And why is it important? Why are microtubules important? It makes up the cytoskeleton, which determines the shape of the cell. So here, I've summarized the function of microtubule. It is structural support, so as I said, the cytoskeleton, and there's transport. So vesicles and organelles can be moved along the microtubule. And when I say cell division, the centrioles, so microtubules make up centrioles, and these centrioles form these things called spindle fibers in which chromosomes line up during nuclear division. Speaking of centrioles, here we are. Centrioles and centrosomes. They produce spindle fiber during cell division, which you will cover in AS actually during your cell division topic. Now to break it down and make it simple, 2 centrioles makes 1 centrosome. And each centriole contains 9 triplets of microtubules. Wow, that's a mouthful. So here, as I said, you can see centriole, but you can't see a microtubule. So here you can see a microtubule triplet. So very small things. And finally, guys, we made it to plant cell structure. It means we're almost done and almost there. Now, what do plant cells have? Most of these things are pretty much what we have covered, except for chloroplast and large permanent vacuole and cell wall. Now, these are the 3 things that a plant cell has, which an animal cell doesn't have. And things which I mentioned, such as centrioles and microvilli, plants don't have those. So let's go ahead and check out chloroplasts. So here is a more detailed structure of a chloroplast. Excuse me. So chloroplast, again, is very well suited for its structure, just like the mitochondrion. And again, just like the mitochondrion and the nucleus, it is a double membrane. Now the chloroplast's function, the thing that you need to remember, which is most important, it is the site of photosynthesis. The rough size is around 3 to 10 micrometers, so you can see it pretty clearly with an electron microscope. Now, thylakoids are these discs here, and thylakoids, so stacking thylakoids makes up this thing called grana. Okay, so this grana or granum, aka stack of thylakoids, they contain pigments which absorb light and electron carriers. So during photosynthesis, you need to absorb light. So different pigments absorb different frequencies of light, and they're all located in here. So this is where the main magic happens in photosynthesis. And there will be other things such as lipid droplets and starch grains, which will be like little dark circles in diagrams, which is not on this diagram, but it will be. So if you see dark circles in chloroplasts, you can probably guess and say that they're either lipid droplets or starch grains. And then the fluid inside, so aka I guess you could say the cytoplasm of the chloroplast is known as the stroma. That is the interior fluid in the chloroplast. And as I mentioned, it's very well suited for its structure. So just like the mitochondria, it has a lot of enzymes and things like that. And also, just like the mitochondria, scientists believe that chloroplasts were independent creatures until the plant cells colonized it in this case, because animal cells don't have chloroplasts, because they have 70S ribosomes. So here, let me just point out on the diagram, the chloroplast is here, this little thing right here. And now we have two more organelles, I guess, to cover. The first one is the cell wall. Animal cells do not have cell walls. We only have a cell membrane. The cell wall's function is to protect the plant, the plant. Yeah, I guess the plant, but more importantly, the cell. So there's the cell wall, which if I can find here, the cell wall, and then there is the inner membrane, and then there's the cytoplasm. So the cell wall is a protective barrier, and more importantly also it's used for structure and support for the cell, because it controls the shape of the cell. When water enters and leaves the cell, it makes sure the cell doesn't get too full and that it doesn't burst, so it maintains the shape of the cell. And another thing is a large vacuole, so it provides structural support. The large vacuole animal cells do not have. Our vacuole is temporary. It shrinks and it leaves depending on how much water we have in our system. But a plant cell will always have a vacuole in its cell, no matter whether it's shrinked or not, but there will always be a vacuole present. And its job is to provide structural support, support and growth, storage, waste disposal, and protection. So numerous functions there. And that is it, you guys. We have covered actually all of the organelles which you need to know for your exam. Now let's check out the summary point. The first summary point, it distinguishes between prokaryotic and eukaryotic features. It is a table summary. I'll just give you a second to note it. Good, now that you've print screened it, take this table down and memorize it. It literally summarizes what I've been talking about. And then moving on to summary point 2, I'll give you a second to screenshot each slide. So that is summary point 2. It basically lists all the organelles which we have discussed about and tells you whether it's in an animal or plant cell or in a prokaryotic cell. It gives you the function and it also gives you a description. So I hope you found this video helpful. If you did, please, let us know down below. If you have any questions, please do comment it down below. We'll do our best to answer your questions. And thank you guys so much for watching. See you!